biomarker efforts. So yes, after every presentation, there'll be an opportunity to ask a few questions. I'll try to moderate those through the chat and capture them and ask them. If those questions don't get addressed in that moment, we hope to have an opportunity to join with a lot of the folks who are presenting during a roundtable session in the afternoon today to have those questions asked. So starting the lineup this morning will be Dr. Katie Planey, who's CEO at Acelot. I hope I said that correctly. Dr. Planey will be presenting on novel small molecules that reduce TDP-43 and other amyloid-related pathologies. And with that, I will turn the Zoom over to Dr. Planey.
Great, thank you. Okay, so just confirming we're good to go. You can see the screen. Okay, great. So I first want to thank ALS ONE and MGH for the opportunity to give this talk. And of course, for everyone else listening today, in particular, the patients and loved ones of patients. So in brief, as an executive summary, Acelot has a platform with first-in-class oral small molecules for neurodegenerative diseases. This is powered by a machine learning platform that was invented by our founder, University of California computer science professor, Dr. Ambuj Singh. Acelot's development candidate ACE 2023 for sporadic ALS disrupts misfolded cytoplasmic TDP-43 and also restores functional nuclear TDP-43. ACE 2023 is showing target engagement against human brain-derived misfolded TDP-43 from ALS, FTD, and Alzheimer's patients. We have our full non-GLP data package completed, and our CMC development batch is underway. So we're estimating and projecting that we'll start our IND-enabling studies for ACE 2023 in mid-2024, and that we would begin phase 1 clinical trials in mid to late 2025. So our leadership team has decades of drug discovery experience. As already introduced, I'm Katie Planey. I'm CEO of Acelot. I'm a leader in building AI and ML-driven biotech companies. That's from my PhD background. I received a PhD in biomedical informatics from Stanford. Prior to Acelot, I co-founded an exosome therapeutics company, MantraBio, and I led the research there for a good 6 years. In addition to our fabulous co-founder, Dr. Singh, a UCSB professor, Acelot is also led by Lewis Whitehead, our VP of Discovery, and Vidya Mathur. I also want to highlight our wonderful preclinical team because you'll be seeing a lot of data that they produce today. And so that is Dr. Eric Shao, Dr. Marcella Coates, and Dr. Shruti Arya. We do collaborate with recognized experts in neuroscience and ALS. David Bleekman, the former CSO of neuroscience at Eli Lilly, is our formal advisor, and we do collaborate with a lot of amazing researchers in ALS, like Sami Barmada and Jean Yao. We also consult with Aaron Gittler. I do want to express a lot of gratitude also to Niels. We recently have begun to start having clinical advisory meetings with them, and we greatly appreciate groups like ALS ONE and Niels that can help guide early stage companies like Acelot to make sure that we're set up for success as we get to the clinic to make sure that our therapies actually reach ALS patients. So as I mentioned in the executive summary, we're aiming to be in phase 1 clinical trials in mid to late 2025. I know that if you're an ALS patient or you're a loved one of an ALS patient, that probably can't come soon enough. I really want to assure you that we're moving to the clinic as fast as we can. Our platform reduces time to development candidates with lower cost and headcount. So we actually raised a seed round just last year, and in just over 12 months with 8 employees, we've accelerated to have this development candidate, and we're preparing for those IND-enabling studies. Acelot takes a very different approach to targeting misfolded proteins. I do really want to emphasize our rigor here. We do have an in-house lab here in J Labs in Johnson & Johnson's incubator in South San Francisco, California. We also really emphasize direct measurement of target engagement, that is misfolded TDP-43. We also measure very specific targeting of the certain size ranges of misfolded TDP-43, and we make sure to validate that we have a functional restorative mechanism of action. I'll be showing you data on all of that today. So our compounds are optimized to target functional misfolded proteins and restore normal homeostasis. TDP-43 is just one of many of these misfolded functional proteins. An appropriate mechanism for these proteins is not clearing the protein, because a lot of these proteins are quite important for normal functioning, but you basically want a functional restoration where you restore the normal functioning protein. I just want to point out that there's been a lot of exciting news in another type of misfolded protein, beta amyloid, recently, that also forms an aggregate like TDP-43, but it is a pretty different protein. It's not functional, and so a mechanism that is very appropriate as a beta amyloid therapy is clearance of large amounts of protein. Acelot does have assets across multiple targets and diseases, but of course today I'm focusing on our development candidate ACE 2023 that will be applied to sporadic ALS as our first indication. So digging a little bit deeper into that development candidate, ACE 2023 treats both the TDP-43 gain and loss of function pathways in ALS. Everyone on the call will hear a lot about TDP-43 today. There's certainly many other companies that are also targeting TDP-43, but given Acelot is the first company of the day, I just want to give a little bit of background on that target. At Acelot, we're incredibly excited because at the pace we're progressing, ACE 2023 could be the first in candidate that is targeting TDP-43 very directly, and I'll show some data on that. But in brief, in its normal state, TDP-43 is located in the nucleus. It does perform vital RNA binding activity, so you definitely don't want to simply remove TDP-43. And in ALS, TDP-43 is lost in the nucleus, but then there's a gain of function where it moves into the cytoplasm, and you have a gain of misfolded cytoplasmic TDP-43, and that results in neuronal degeneration. Small molecules are a very appropriate modality for targeting TDP-43 misfolded proteins and restoring function. It's been wonderful to hear all the talks yesterday and the talks we'll hear today about the different modalities. We do need different modalities and different therapies to treat ALS. I just want to point out why, for the type of function that we're targeting, small molecules can be a very appropriate modality. In terms of TDP-43 as a functional misfolding protein, it's intracellular. It is driven by multiple pathways. We all know ALS is multifactorial and a very complex disease. And different conformations for TDP-43 can differ even across ALS patients and also across different patient populations. TDP-43 is also found in FTD and Alzheimer's, for example. Acelot's compounds are highly cell and brain penetrant. They directly target these misfolded aggregates, and they reset homeostasis to treat all of these upstream causes. In particular, I'll show you some data today that our compound is effective across different conformations for TDP-43 found not only in ALS, but also FTD and Alzheimer's disease brains. So our development candidate, ACE 2023, restores the functioning nuclear TDP-43. So in brief, this is a picture of a motor neuron cell membrane. And ACE 2023 has very high cell and brain permeability. So essentially it enters the cell, and in the cytoplasm, it disrupts these disease-associated misfolded aggregate seeds. And that essentially pushes back the protein dynamics and equilibrium into functioning nuclear TDP-43, which is in monomers and dimers. ACE 2023 does directly engage the misfolded TDP-43. And so it also restores the functioning nuclear TDP-43 isoforms. One drug molecule can sequentially disrupt multiple aggregates. That's because after ACE 2023 disrupts that aggregate equilibrium, there is no more target for it to engage. So it can go ahead and engage with another misfolded TDP-43 target. This is just a picture of ACE 2023 engaging the center core of a misfolded TDP-43 cylindrical hexameric aggregate. That just means that that's 6 TDP-43 monomers essentially clumped together, right? And that's its misfolded state. And this is the example ACE 2023 small molecule, and green is just showing those areas of engagement where it's engaging the center core of that aggregate. ACE 2023 specific engagement with misfolded TDP43 is confirmed by multiple high-resolution assays. So on the left is an atomic force microscopy assay. So you basically get to look at nanoscale at proteins. And on the far left-hand side is misfolded TDP43. The misfolded TDP43 is actually the smaller dots, the puncta. And so when you treat with ACE 2023, you see that we completely remove that misfolded TDP43. On the right-hand side is an assay called ion mobility mass spectrometry. What's important about this assay is it does let us really interrogate the specific size ranges. As I mentioned earlier, monomers and dimers of TDP43 are functional, so we do want to make sure that we're not decreasing those size populations. This is a recombinant cell-free assay, and so you add misfolded TDP43, and you can see that it tends to form this hexameric misfolded aggregate. After you add ACE 2023, it dissolves that misfolded aggregate, and it actually starts to restore those functional monomer and dimer levels. And that was in a cell-free assay with just recombinant TDP43, but I want to emphasize that we're seeing this same trend in human ALS brain tissue derived misfolded TDP43, which is quite exciting, which means that ACE 2023 does restore the TDP43 homeostasis in ALS human patient brains. This is an experiment run with Dr. Raquez Kayed at the University of Texas Medical Branch. He's a well-renowned expert in protein aggregates. And so essentially on the left-hand side, you're seeing these human ALS brain tissue derived misfolded TDP43 aggregates. This is a seeding assay. And essentially when you add ACE 2023, you see now that essentially you're seeing smaller puncta, right? Which what that means is the functional monomer and dimer TDP43 isoforms are being restored. ACE 2023 also restores TDP43 loss and gain of function mechanisms in cellular assays. So I'm going to walk you through a few of those. On the left-hand side, for everyone in the ALS research community, this will be a very familiar assay. This is your basic stress induction assay where it's a TDP43 overexpression system. And so the small dots are misfolded TDP43 cytoplasmic aggregates, and then the larger circles are cell nuclei. That's non-toxic, right? That's the functioning nuclear TDP43. And so when you treat with ACE 2023, you see that we remove those misfolded cytoplasmic aggregates. This assay is great to confirm that you are resolving that toxic gain of function, but you do need a non-overexpression assay to understand if you're actually restoring the nuclear loss. And so that's this second figure here, is that this is a stress-induced cell system, but it's not using TDP43 overexpression. And so this is data that we recently collected that we think is very, very exciting because we're showing in a dose-dependent manner that we do reverse the TDP43 nuclear loss, meaning that as you increase doses of ACE 2023, you see an increase in nuclear restoration where you're almost completely restoring the levels back to a non-stress state. We have also conducted studies in ALS iPSC-derived motor neurons. This is in collaboration with Dr. Sami Barmada at the University of Michigan. Essentially, in this experiment I'm showing here, we dose these iPSCs a single dose of 300 nanomolar on day 1, and then you essentially just track the cell survival for 10 days. There's no media change, no nothing. And we're seeing that we increase survival by 35% in these ALS iPSC-derived motor neurons. And these iPSCs are derived from ALS patients. And so we do find that quite exciting in terms of emphasizing that our compound is having a restorative mechanism. So, of course, those were cell-based assays. You obviously need to be validating in animal models also. ACE 2023 demonstrates target engagement and functional improvements in a very aggressive iTDP43 A315T mouse study. So essentially, the design here was that we started dosing the animals at 4 weeks to ensure that they were already showing ALS pathology. We think it's very important to start treating when the pathology is already developed because that will be the real case of ALS patients in the clinic. And then we did daily oral dosing for 8 weeks. ACE 2023 is highly orally bioavailable. So in terms of brain PK, we're seeing very high drug levels in the brain. So that's great in terms of target coverage and potency. In terms of target engagement, we are seeing a nice reduction in TDP-43 in the mouse brains. In terms of efficacy, we're seeing an improvement in motor function and also a decrease in cortical neural inflammation, which includes the motor neuron cortex. I want to double click on some of these in vivo assays, and I'll do that in the next couple of slides. So ACE 2023 demonstrates target engagement, i.e. that misfolded TDP-43 across multiple readouts. This is what we're really excited about, is that we're seeing consistent trends across multiple readouts and ways that you can measure TDP-43. This highlights our emphasis on rigor again. Many animal studies don't actually directly measure TDP-43 aggregates, but that is our direct target. So we collaborated with Dr. Yatsi K at Macquarie University, who did a wonderful job of helping us really interrogate and confirm our target mechanism. So we measured TDP-43 aggregates, phosphorylated TDP-43 aggregates, ubiquitinated TDP-43, and cytoplasmic TDP-43. We're evaluating right now with our future clinical plans on whether phosphorylated TDP-43 could be a viable treatment response biomarker, given the strength of response that we're seeing in this animal model. And I do want to note that this animal model has orders of magnitude more target load than you'd actually see in a patient. So we do find these trends very encouraging. In terms of functional improvements, ACE 2023 does demonstrate functional improvements across multiple readouts. This is a motor pole function test. Essentially, you're letting the mouse climb down a pole. And the number of mice who can complete that test increases when we dose with our compound. And then in terms of an anxiety behavior elevated plus maze, you see an increase in the time spent on the maze. And we are also still analyzing data from this study, and we're beginning to see also improvements in neuroinflammation markers, which is also quite exciting. So I also want to note here that that was in a lot of very ALS-specific models, but we're very excited that ACE 2023 can treat different TDP-43 target conformations found across quite a large number of patient populations. To be clear, our entry indication is sporadic ALS. There is such high unmet need there, and we have run most of our validation assays in sporadic ALS. But quite a few other patient populations do have TDP-43. And so we conducted a study where we looked at human brain-derived TDP-43 aggregates in Alzheimer's, ALS, and FTD, and that's on the right-hand side. And so essentially in the first column, you're just looking at PBS, it's a negative control. The second column is TDP-43 aggregates. This is using a polyclonal TDP-43 antibody developed by Dr. Rakesh Kayed. And on the right-hand side, you can see when we dose with ACE 2023. So we're very excited, in particular in ALS and FTD, you're seeing a near complete removal of this misfolded TDP-43 protein, these aggregates, and you're seeing quite a dramatic reduction for Alzheimer's patients also. So this is just very exciting that we can be reaching a large number of patients, and it emphasizes the benefit of the small molecule mechanism here because we can engage with slightly different target conformations. I also want to note, since we're well on our way to the clinic, that ACE 2023 does have very desirable PK, ADME, and safety properties. Just to call out a few, we're seeing greater than 70% free brain fraction with oral route of administration. We're not seeing significant drug-drug interactions, meaning CYP interactions. We're not seeing any cardiotoxicity. We're not seeing any significant off-target hits on panels such as urofins, and we're not observing any mutagenicity and no genotoxicity. As I mentioned in the executive summary, we have completed our full non-GLP ADME package. We are actually submitting this week our Type D meeting request with the FDA to share all of this data and kickstart our path into the clinic. So ACE 2023 has a clear biomarker and clinical trial strategy. Of course, like most ALS companies today, we certainly will be measuring neurofilament light chain in the clinic, but our animal studies have suggested additional potential biomarkers for treatment response, such as phosphorylated TDP-43 and GFAP in mouse models. And so we're looking at beginning to develop those biomarkers because, as we know, biomarker development has also slowed some therapeutic developments in ALS. As I mentioned, we're submitting the Type D meeting this week. We're aiming then to run the IND-enabling studies next year, and ideally enter then into phase 1 clinical trials in mid to late 2025. In terms of our clinical trial design, we're following the best practices that we've seen other companies do to date, where you do have your phase 1A in healthy patients and then a phase 1B, and then potentially an 18-month dose extension. So I just want to wrap up the presentation to say that we're incredibly excited that we're seeing that ACE 2023 does essentially target both the gain and loss of functions in TDP-43. We're very excited by the compound's druggable properties, and we're moving as fast as we can to get it into the clinic so that we can finally get it to ALS patients. And with that, I would be happy to answer any questions.
Thank you, Katie. That was a fantastic presentation. Thank you for sharing so much data and so much about your plans. We do have a couple of questions in the chat. One of them you largely addressed during your presentation, looking at TDP-43 pathology, not only in ALS, but also in other diseases. There was a specific question about VCP disease, and I don't know if that's something on your radar.
I want to be honest that it's not, but this is why this is a great symposium, because I would love to connect with that person. There are some other small rare diseases that clinicians have suggested, but I would love to learn more about that, because we want to be very mindful in our clinical trial strategy that we're not leaving other patient populations out.
Thank you. And another question. About the affinity of AC 2023 for TDP-43. Is it a micromolar or nanomolar range? And then there's a follow-on question as well.
Yeah, that is a good question, because it is a different mechanism than a high affinity antibody. So it's essentially the small molecule essentially engages the aggregate with multiple weak binding interactions. The way we do confirm that is through molecular dynamic simulations. We work with the Jones Shea group at University of California, Santa Barbara. As many of you may know in the research community, there are not really accurate crystal structures of TDP-43. So that is the way that we've confirmed the mechanism. That is also why the compound doesn't stay with the aggregate once it dissolves. It's a one-to-many interaction. So when it dissolves the aggregate, it can move on to the next aggregate.
Thank you. And maybe you didn't measure, but is there a survival advantage in the mouse model that you've studied?
We have not measured it yet, simply because we had to sacrifice the mice to get the target engagement. It would be a different animal model to measure survival. And so we're looking into that with one of our collaborators, Yahtzee at the moment, but we have not measured it today. All of the mice did survive in the study, I will say, we were excited. There was no toxicity or anything.
Thank you. And there's been a lot of focus in the field in recent months and years around missplicing as a function of TDP-43 loss of function. Is this something that you've looked at, and are you looking to see if the compound recovers any of that function?
Yes, we are actively looking at it with a collaborator right now. We're encouraged that we're seeing the nuclear restoration. I think then the question is there's several missplicing metrics, right? Like, which one will we see? And that is what we're measuring over the next few months.
Fantastic, thank you. And I'll take the prerogative of a question of my own. It seems clear that the molecule does disrupt the aggregates that form. Does it also prevent the formation? Does
Yes.
it work both ways?
Yes, that is a great question. We do have data showing in a cell-free assay where, in a nutshell, it does. And we have proved that through, essentially, it's called the THT assay, but you can add the compound at the beginning, or you can wait for aggregates to form and add the compound. And we see a very nice reduction in aggregates in both of those situations.
Fantastic. And I think since we got started a little early, you're getting a lot of questions because we have
That's
time.
fine. I'll take it. Yeah.
But we've got I'll take one more. Um, do you have thoughts you can share with us on clinical target engagement biomarker?
Yes, yes. And so I guess I will clarify, since I can't directly talk to the person, I know there are treatment response biomarkers and biomarkers for accelerated approval. Pragmatically, I think most of us in the ALS community are focusing on NFL. This mouse model does not show a strong increase in NFL with disease pathology. So we are looking at exploring that in cellular models and animal models. But regardless, as a small company, really the only feasible clinical response biomarker is NFL for accelerated approval. That just takes years of work with the FDA and a lot of money. But in terms of treatment response, yes, we're looking at NFL, but we also have very preliminary data. I didn't show it here, that we're also seeing a really nice trend in GFAP and EPA1, along with the phosphorylated TDP-43. So those are the other biomarkers that we are beginning to then work on developing and validate in human samples in-house that we would bring into the clinic.
Fantastic. And I fibbed. I have one more question
Yeah,
from the
fine.
chat. You've shown in the iPSCs from sporadic ALS patients the effect. Does it work in the context of ALS linked mutant TDP-43?
I want to note that actually was a mutant ALS iPSC motor neuron. It was in short, iPSCs from mutant patients with a mutation do show a stronger pathology. And so in research, we tend to use the ALS patients with the mutation. So yes, in a nutshell.
Well, fantastic. Thank you very much. I hope you'll be able to join for the panel later. And if questions weren't asked, you'll have an opportunity to address them later.